Good evening, good morning. Yeah, it can be good evening, good morning, anything. All good. Right. So, yeah. So we start with this very interesting topic. Right? This is synaptic transmission. So I just got the news from you. Chirag, I read your mail bit late, but as you mentioned that now currently the lecture is going on. So chances are very high that right now also there will be less students will be able to attend things live. Anyway, my I was really feeling very much that this particular session, if it is attended by more students live, it would be very beneficial. Anyway, I read your mail a bit late. Okay, so that's our purpose for the synaptic transmission. That's the topic for today, right? So if we divide at a very basic level, right? Because same thing will be carrying on to into the pharmacology, right? So if we say that that's our body, and in our body it is divided into, say, there is a CNS, there is a peripheral nervous system, and then there is, say, lecture is up to only 5 p.m. university lecture. Okay, okay. But by the time we'll reach to at 5 p.m. so we will be covering lots and lots of ground. Anyway, chalo again, right? So we just write it like this: CNS and PNS, right? Or we can make it like ANS, autonomic nervous system, right? That would be much better. So here we have got parasympathetic, right? Parasympathetic, sympathetic, and the enteric, right? Enteric nervous system. So this is rest and digest, and this is for fight and fright. In case of enteric, it would be a combination because this is in GIT, right? So it would be a combination of parasympathetic and sympathetic. Our today's focus would be on this, say parasympathetic. Say because of the topic of today, neurotransmitter, we want to talk about acetylcholine, right? That's normally what we say, that acetylcholine, that means parasympathetic system. No, but we'll try to clarify certain very interesting facts before we really proceed, right? So when we say, let's say that's our brain, right? That's our brain, and then there is the spinal cord, right? So this is cranium. So that is cranio, and this one is sacrum, that is sacral. Craniosacral, that gives parasympathetic. That gives parasympathetic. And over here, that is thoracolumbar, right? It is thoracolumbar. That is what is giving us sympathetic. To be more specific, when we talk about parasympathetic, remember this number 3, 7, 9, and 10. When we'll be discussing in detail about the cranial nerves, at that point we'll be coming back to these numbers. But remember, this is cranial nerve number 3, 7, 9, and 10. They'll be playing major role, right? Major role into the parasympathetic system, right? So these are all cranial nuclei, right? Cranial nuclei. When it comes to, say, sacrum, remember S2, S3, S4, right? This much is necessary. For thoracolumbar, it is very easy. All the way from T1 to S1, right? T1 to S1. This is a catch. right? Because we said sacrum, but yes, S1 is part of thoracolumbar, that is sympathetic, right? More when we'll be discussing the anatomy of abdomen, right? As such, it has already been discussed and it is into one of the lectures on our channel. But this is the thing, right? This is the basic. Let's go into the brain, right? So that we make ourselves crystal clear. In brain, there is limbic system, right, which handles all those emotions, right? That means, I mean, everyone is feeding to hypothalamus, right? So this is the main command center, hypothalamus, right? Let's draw hypothalamus something like this, so which will tell us like this, right? So this one is anterior and this one is posterior, correct? So obviously this one would be lateral. So entero, enterolateral, right? Enterolateral part of hypothalamus, that is parasympathetic, right? That is parasympathetic. So this one, parasympathetic. And the posteromedial, right? Posteromedial part is sympathetic. Fair enough, right? So this is sympathetic, fire, aggression. So we keep it red. And this is rest and digest, right? So this is like, let's put it green. So this is our hypothalamus. So hypothalamus obviously extremely important, right? For your memory, learning, emotions, everything, right? So main functions like your memory, emotions, learning, right? Learning, very, 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 very vital. Drawing again, that was the section of spinal cord, and over here, right, that was our ventral root, right? This was our ventral root or the anterior root. From anterior root, there was one ganglia, right? And then it was merging and forming the spinal nerve. But this was the ganglia, right? This was the ganglion. This ganglion, there would be few fibers which will be starting from here and they will be going to respective organs. So that means there would be few fibers which will be before that, correct, before that. And we called them, these fibers, as preganglionic, preganglionic, and these fibers will be postganglionic, correct, postganglionic fibers, right? So this is preganglionic and that one, these are the postganglionic. That means this particular ganglion, right? That is very vital. Something very interesting is happening at this point, at this point. So we can, in nutshell, we can write it like this, right? That's our ganglion. And before that, it is called as pre, if the direction is this, and this one is called as the postganglionic. At ganglion, there would be neurotransmitter, right? That's how they will be talking to each other, right? They will be communicating because there is a gap, and that's where the synapse is formed, right? That's where the conduction occurs. So it means from here, the action potential, this is general visceral efferent. Absolutely perfect. Absolutely perfect. This is general visceral efferent because this exit is efferent. And this ganglia, it takes part in only the visceral, general visceral. So you are very right, right? General visceral efferent. Because somatic, for somatic, this ganglia will not come into picture, right? Achha. So this is the neurotransmitter, right? And action potential is like an electrical signal. And over here, neurotransmitter is like a chemical one. From chemical, once again, it will be converted to the electric one and things will continue, right? So far, so good, right? Everything is fine. if we go into slightly more details, right, it is not so that they are like connected very honestly, right? That means that this is how we really draw, right? That's the exon, and these are the, say, dendrites, and then that dendrite would be connected to these terminals. They will be connected to another dendrite, right? And that's how, like, very in a very disciplined way, all the neurons are connected like a train. No, it's not like. It can, the another neuron, it can be connected over here like this one also, right? It can even, like, head to head, it can get connected like this also, right? All possible combination. So it's like that exon, it can get connected to straight away soma, the cell body, so then we call it exosomatic, right? We can call it exosomatic, right? If exon to exon, so it is exon, right? Exon. Or if it is exon to dendrite, so we can call it exodendritic, right? So that means all possible combination. That's how the complete network is formed, right? But one simple thing, whatever is happening, and whatever the level it is happening, no, right? So it is that's why the conduction would be, that's what we are coming to. Just give me a minute, right? The conduction would be always going, right, in the direction like this, right? Yeah, actually, I think we should remove this, right? Not like this, but say exon to exon, right? Exon to exon, that is there. Exon to dendrite, exon to soma. Yeah. So this can be like this, say like this. Okay. Huh. So what I was telling, that all these things, they are occurring because of neurotransmitter, and there are more than 100. Yeah, actually, there are more than 100 neurotransmitters. That's why these names will keep on coming again and again. So these neurons, they are grouped as per their chemical structure, right? Very simple, right? So they are called as the class. They are called as the class of neurotransmitters, and pretty decent. The thing is, one is like they are amino acids, right? They are amino acids, and in amino acids you'll find the very famous glutamate and the GABA, right? Gamma amino butyric acid, right? It is gamma amino butyric acid, butyric acid. So when they say that pharmacology is very complex, no, it's not very complex. It happens like that. Once we understand that this GABA is inhibitory, GABA is inhibitory, glutamate is stimulatory, right? So what exactly it means, stimulatory or inhibitory? Simple as that. As we learned that if this is the cell, and if anything is happening, if the inside of the cell is becoming more positive, that means it is likely to go for the action potential. ठीक है, right? So here is a channel, and this particular channel is what, this particular channel, right? This particular channel is sodium operated, voltage gated sodium operated, or ligand, whatever. But this particular glutamate, right, when it would come, it would open this particular channel. So what will happen, this sodium is going to rush in, sodium is going to rush in. That means there would be more positivity inside the cell. That means this cell will be going for action potential. Done, right? When it will come to glutamate, GABA, right? GABA. What GABA will do, right? GABA will GABA will also open some channel, right? GABA will open some channel, but this time it is opening the which channel? Chlorine channel. So that chloride is entering, right? So negative, 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 negative. That means this thing won't go for action potential. That's how they operate. So easy, so simple, right? So bus this is what we have to understand. And today we'll be going in depth about, because now we'll understand that why acetylcholine is so vital. At so many places it is really acting, right? So this is about amino acid. We can also add one more G, glycine, right? So all of these, they are amino acids, right? The second group, if we see the second group, that is nucleopeptides, right? Nucleopeptides. And nucleopeptides, very important because they release endorphins, right? Endorphins. Very important, right? Because these endorphins, they give that amazing feeling, right? Another important one, monoamines, right? So almost just consider now whatever the rest of the neurotransmitters you remember, all of them, they will be part of monoamines, right? Whether we call it, say, this epinephrine, right? Then norepinephrine, right? Norepinephrine, then happy hormone, serotonin, dopamine, right? Dopamine, serotonin, histamine, right? Histamine, all of them, right? These are neurotransmitters, right? So all these will come one after the another, right? As we'll start discussing the physiology more and more. But the fourth and the most important part is the acetylcholine. This acetylcholine is our chief guest for today, right? Acetylcholine. Sodium and chlorine, will it burst? No, it won't burst. No, it won't burst. Because when there is, at one point, it is never so that sodium will keep on coming in and in and in and in and in, right? It will never happen. At one point, yes, you are very right, that there will be a balance, right? A stage would come when the reverse cycles would start and things will try to come back to its original status. And yeah, sodium potassium pumps, they are always there, which keep on working, right? So cell will never burst. Achha. So this is acetylcholine, right, which is technically called as the ester of choline. Right? So we'll be discussing about acetylcholine in really good depth. Achha. Now, if we talk about a very, very simple thing, right? Because then we'll elaborate on it. So here is our synapse, right? So this one is pre-synaptic and this one is post-synaptic region. And because this is our synaptic cleft. Few things, that all these neurotransmitters, they will be in the form of bags, right? These are vesicles. These are vesicles. And these vesicles, they are all connected with each other, right? They are all connected, so that none of the vesicle, it can really get free and reach to the surface. No, there would be a very specific mechanism. So this is synaptic vesicles or the vesicles, and they contain neurotransmitter. So in our case, it is our acetylcholine, it is there, right? So in nutshell, what would really happen is, as soon as the action potential is going to come, as soon as action potential comes, that action potential will be working on the sodium channel, calcium channels, right? It will be working on calcium channels, calcium channels. So these calcium channels, these ligand gated calcium channels, they will open and large amount of this calcium is going to rush in. And the entire game, that is, from this point onwards, is the game of calcium, right? So this is in nutshell what I talked about, that what would really happen. Because then this vesicle, they will all break. This vesicle will come to the surface, it will break, right? What is called as the exocytosis, and then it will release those neurotransmitters. Those neurotransmitters, they will reach to the receptors, right? They will reach to the receptors. Now, acetylcholine, it will work on what, right? It will work on what? Well, it depends upon on which receptor it is working. So don't get confused. Acetylcholine is released over here, right? Acetylcholine, right? It is released, but depends on receptor it works on, right? That means we need to know something more about these receptors also, right? So, but this part is clear that it is the calcium channel which is important when the action potential will come. Still, there is a mystery how acetylcholine came into these vesicles, right, in the form of bags. From where did they come, right? We'll talk about it, because that is the basic mechanism when we'll be coming to the medicines, that from at what point we really want to stop what. Achha. So we were talking about receptors, right? So now this is acetylcholine. It will work. Acetylcholine as such is parasympathetic, right? Parasympathetic. And in parasympathetic system, we have talked about that there are two types of receptors. One is what is called as the nicotinic receptor, and second what is called as the muscarinic receptor, right? So this muscarinic receptor and the nicotinic receptor, put it like this is stimulatory and this one is inhibitory. But inhibitory to what? Where they are present, right? So this nicotinic receptor N, right? It is at neuromuscular junction, right? Neuromuscular junction, right? Yes, vessels will come. Definitely vessels will come. So, do the vesicle come from Golgi? Yes, will come. We'll see. Yes, it would be like that. But yes, we'll talk about it. Just few minutes. So, neuromuscular junction, right? So, because it is stimulatory, that means it will contract. It would contract, right? So, this leads to contraction of the muscles. When it comes to muscarinic, right? So this muscarinic is inhibitory, right? And it is inhibitory to what? Cardiac muscles. Cardiac muscles. So over here in GIT, yes, they will contract and they'll start the process of digestion, right? When it comes to cardiac muscles, so it is inhibitory to them. So it will slow down, right? It will slow down the heart, right? It will slow down the heart. So typical rest and digest phenomena. Right? So that means we need to know that where exactly these typical receptors are. But it's not so that this once acetylcholine is released, so bus it will keep on working on and on and on. No, its action is only for a very short duration. That window is very short, just few milliseconds, right? Action is only for few milliseconds. So there would be two things. Again, we'll be watching it in more detail, right? But this I'm giving you a bird's eye view. That this acetylcholine, when it is there, it is necessary to take it off. We don't want that it should keep on working on and on and on. So one is by you degrade it. Degradation of acetylcholine. So we need to break it so that it should not work, right? And second, take it back. Take it back. It is what is called as the reuptake. So this is good, right? We take some of the acetylcholine back, right? If they are trying to act more, so just take them back so that we can reuse it, right? So this reuptake, which is by astrocytes, right? By astrocytes. Plus there is a very good mechanism, right? By the transporter protein. We'll see that. And once again, the cycles, they keep on working. As we proceed further, right, as we talk about, say, these receptors, there is degradation of acetylcholine. Yeah, so that is acetylcholine esterase. Yes, that would be the thing. Achha. We are talking about now, say, this is brain, right? From brain to spinal cord, whenever there is one fiber which is coming out, this would be the preganglionic fiber, right? Because the ganglion is here, so this would be preganglionic, right? And when it is from lumbosacral, I am writing LS, so lumbosacral, so it would be preganglionic, correct? The point is, remember, for the all preganglionic release, there is only acetylcholine, whether sympathetic or parasympathetic. That means the ganglion which we are watching, this would be releasing acetylcholine, whether sympathetic or parasympathetic. So this is acetylcholine. But only in this case, that is when we are dealing with, say, parasympathetic system, right? So acetylcholine is here also. And when it comes to, say, sympathetic system, so over here, the target would be depending upon epinephrine or norepinephrine, right? So for the target only, these are the changes. But otherwise, for all the inner function, internal function, that is over here, right? That is over here, over here, over here. All ganglia, they are working on acetylcholine only. Very important, right? Next important point. In case of parasympathetic, right, the preganglionic fibers would be long. And in case of sympathetic, these preganglionic, they will be short. So what is the importance? Importance is when we'll be dissecting the abdomen, right? Just near this vertebra, we'll find all preganglionic or those ganglia. They will be near the vertebra. So this one, obviously, right? Because these are short. So we'll be able to see those ganglia near the vertebra. While in this case, they will be far. So that's one thing. Another, this would be the postganglionic, they would be longer, right? They will be longer. And who will be the receptors? Well, in case of, say, sympathetic, now here the receptors would be alpha, beta, right? You remember that, right? Alpha 1, alpha 2, beta 1, beta 2, beta 3, all those receptors, right? Sorry. In case of acetylcholine, that is our sympathetic, the receptors would be, we just talked about, they would be muscarinic or nicotinic. Right? They will be muscarinic or nicotinic. This is the importance, right? And as our today's topic is synaptic transmission, so now our focus will be going on that. But yes, there are few exceptions, like, say, it goes to renal blood vessels, so where the dopamine would act, right? So we can write it, right? Say on renal blood vessels, right? Renal blood vessels. As such, the kidney itself is a superb topic. But on renal blood vessels, so when it is the dopamine, right? So dopamine will act and it leads to vasodilation, right? Vasodilation. So when the vasodilation occurs, what would happen when the vessels dilate? So there will be more pressure, right? Ultrafiltration, there will be more ultrafiltration, right? So when they will go, it will lead to diuresis. More water will be able to pass through it, right? So that's the dopamine. Similarly, say the role of adrenaline, right? It is completely different because that is like a hormonal system and which will act on multiple points, right? So that's a completely different topic. So we are not touching it right now. But this part is crystal clear that our acetylcholine is over here, and over here, right? At all these places. So, new page. Here is our soma, or the cell body, or the cell head, or the soma, right? Head, right? So that's our head. This one is exon, and then the dendrites. It is this place where the neurotransmitter is synthesized, right? So here the peptide neurotransmitter, right? Neurotransmitter is over here, right? It is a high molecular weight, right? It is synthesized by this peptide neurotransmitter over here. Then they travel along, right? They travel. And when they reach over here, it actually becomes acetylcholine. Now this acetylcholine, this is a small molecular weight, right? So here peptides, they are high molecular weight, right? They are high molecular weight, and this one is low molecular weight, right? Low molecular weight. Now it's not only the acetylcholine, right? As it comes over here, even the norepinephrine, right? Norepinephrine, that is also a low molecular weight. So we are now crystal clear about it. So let's start with the real synaptic transmission, right? What would really happen as the things would really progress. Yeah, I think students, they might join a bit late. Okay, that's right, peptide break. So that's what now we are discussing. Now see, first thing which we need to know is we talk about some very basic things, right? Say, and that is when we want acetylcholine, from where this acetylcholine? Choline, it comes from nutrition. This is a nutrition. It will come from our food. From our food, what we eat. We'll go to digestive system, and yes, this is food. Acetyl, that is to be made. Right? So let's see how we start with that. Things will start, our journey would start with glucose. Right? We are going to a very granular level. Right? So glucose. Glucose, we know that it is a 6 carbon molecules, right? It is 6 carbon, and this glucose leads to break, right? And that is what is called as the glycolysis, right? Lysis means break. It breaks. It breaks into two parts, right? And obviously equal. 3C, 3C, 3 carbon, 3 carbon. And what we call this, both are called as the pyruvate, right? Pyruvate. Now I know we are now into the biochemistry, right? We'll be touching a small portion of biochemistry, and then we'll once again come back to physiology. But this thing will give the crystal clear understanding that how everything is connected, right? And tomorrow we'll be adding the pharmacology into it, right? Acha. There were 2 3 mails that the question which was asked is it too early to talk about pharmac? Arey man, why do you feel so? At any point, if you feel like कि नहीं, this pharmacology थोड़ा मुश्किल लग रहा है, right? So just keep it on one side, no problem. But even if you hear those words, right, it will your brain will keep on rewinding it, and you'll be more and more comfortable. Right? Very soon, in this week only, I'll be teaching even radiology. Right? Why not? यार किसी ने X-ray दिया. If someone has given you the X-ray chest, so you should be able to speak fluently. कि हाँ, these are the things which can be seen. Right? One complete session only on X-ray chest, because that is the basic which you must know. Right? So coming back to this, 6 carbon broken into 3 carbon, 3 carbon, right? So that is pyruvate. Now this pyruvate, on this pyruvate acts pyruvate dehydrogenase, right? It is pyruvate dehydrogenase. And what it really does, CO2, one carbon out. So that leads to 2 carbon, and that is acetyl. Acetyl. Now the story is not yet over here, right? It's not yet over. This acetyl, it has got one end. This is not stable, right? It has one end, one portion, which is sticky. Something can really stick to it to make it stable. Something can stick to it to make it stable. And there comes the coenzyme A or CoA. This is coenzyme A. So when that coenzyme joins, there is the formation of what's called as the all-time great acetyl CoA, acetyl coenzyme A, right? And this acetyl coenzyme A, it has got one reactive. If I say it has reactive sulfur end, right? A reactive, what's called as the reactive sulfur end. This means this reactive sulfur end is in search of someone that I can just pick that substance, and that is, and over here what this coenzyme A, well, coenzyme A, if you really go into very detail, right, so it is what it is like a combination, right, that there is vitamin B5, and then there is ADP, to that there is a phosphate bond, right, but this one end, right, that one end, that is free, that is sulfhydroxyl bond, right, that is that ion, right, that group is attached, not bond, that group is attached, right, so this group is attached, so we write SH, right, that group is attached, and so acetyl coenzyme, we can say that it has got this SH, and to this, from our food, choline would come. So this choline and this acetyl CoA, they both join to form acetyl choline, right? This is how it really forms. So this is the whole story. Starts with glucose, glycolysis, 3C, pyruvate dehydrogenase comes, right? One carbon knocked off, two carbon, that's what we call as acetyl, right? Just acetyl. And this acetyl, it has gone one sticky end. So that coenzyme, it comes in the form of a sticker. It connects, but this sticker has got one reactive end, that is sulfhydroxyl group. This sulfhydroxyl group, it gets connected, right? It gets connected to this choline and leads to formation of acetyl choline, right? So this is the in background, right, what really happens. So with this concept, let's start with our today's super journey of acetylcholine, right? So the whole process, right? So journey begins. And we'll just write down the keywords one after another. So today we'll definitely revise it, right? Let me keep on saving it also. Today we'll definitely revise it, right? At a high speed, we'll revise it because now the processes, they are crisp clean, and every process is where certain drug would be acting, right? So in just one shot, you'll be knowing so much about so many drugs. So let's start with our journey. First, from where it is starting, mitochondria, right? Mitochondria, that's the place from where the journey is starting. And what it is doing, we are just telling this is releasing acetyl CoA, right? Acetyl CoA, acetyl coenzyme A, right? So now we are inside the cell, right? So we are inside the cell. That's our cell, right? Let's make it big, right? Big. Okay, we keep it like this, right? And we'll keep on extending it. And we give number. So that's our first number, first step. Acetyl coenzyme that is released. How it is released? Well, it is the joint effort of that pyruvic acid or the pyruvate, which is broken by pyruvate dehydrogenase, and it leads to acetyl CoA. That's the process, right? So we just write as seen above, right? So that keeps this area neat. So as we discussed about it, it comes over here. And the second step is, I think we should write 2 over here, right? 2 over here. From our food, right? So from our food, right, it goes into the circulation, right? Systemic circulation, whatever we eat, right, through the blood vessels, and finally it is the choline. It is the choline, right? And that choline is outside, right? Choline is not in cell. So there comes another channel, right? And that channel is a co-transporter. Now you know what is co-transporter. Co-transporter means that sodium it is coming and it is I'm speaking what came in my mind. Sodium is coming in and sodium is pakading half of choline, right? So that is co-transporter. I usually remember it like that, right? So that's a very nice English, right? Sodium pakading the hand of choline, right? And sodium choline co-transporter, right? Co-transporter. And it takes choline in, right? So now choline is inside. Choline is inside. Now this choline, बस वो तो तैयार ही बैठा है, right? Acetyl CoA, it has got that reactive end, right? So we give this sodium choline co-transporter as number 3, and acetyl CoA and choline, they both meet, and now we have got acetyl choline, and that's the number 4. So material is ready, right? Now this acetylcholine is positively charged. It is positive. So let's come to now. This is not in proportion to the cell, but this is our vesicle, because so many things will be occurring in this, right? So this is our synaptic vesicle. There would be several such vesicles. Sorry. There'll be several such vesicles, right? But this particular vesicle, there would be again a channel, right? Again a channel. So let's open this channel. This channel is a very classic channel, right? There is this acetylcholine goes inside, and in order to balance it, hydrogen will come outside, right? They keep on working. So gradually, slowly and gradually, there is built up of acetylcholine over here. But as you know, there is always a control mechanism, right? There is always a control mechanism. There would be ATPs, right? There would be ATPs. So these ATPs, they are negatively charged, right? They will be negatively charged, so they will be attracting, right? Similarly, there will be HSG, right, which is negatively charged, right? This is heparan sulfate. These are all glycoprotein, right? Let's write it somewhere. We write it HSG here, right? HSG. It is heparan right? Not heparin, sulfate, and these are glycoprotein. Glycoprotein. So they are also there. So these are negatively charged, right? So they will attract, right? They will attract, and thus our vesicle is now getting loaded with acetylcholine. Where exactly? Which number are we? Right? So acetylcholine, that was number 4, right? So it is formed, and number 5, that comes to the entry, right? Or yeah, or let's put it this way, right? Let's put it this way. Say choline enters. This is number 4, right? Yes, through channel just now. So 4, and this is number 5. Acetylcholine is produced, and through this channel, right? Through this channel, which is already there, right? Acetylcholine enters in, and this comes out, right? So far, all good. Let's introduce more vesicles, but we'll make them small because they are so many, right? So, and all of them, they are connected, right? Let's not draw it on this side, right? We'll use it for some other purpose. Here, right? These vesicles are everywhere, right? So many of them, and they are connected with the cytoskeleton, right? That is with the cell structure, right? They are connected, and this is a very special substance, and that is called a synapsin, right? So to avoid confusion, we just write acetylcholine plus, this is also acetylcholine, right? And we do have that HSG minus and ATP minus. So that's all our vesicles, but they are connected with each other. So they are not left unattended. So that is by synapsin. So it binds vesicles to typical cytoskeleton, right? Cytoskeleton is the space which we have drawn, right? If you remember when we discussed the cell, right? So inside cell is not like a empty like a balloon, right? Completely hollow, right? This is the structure which is inside in the form of fibers, so that skeleton, exactly the same way, the way we have got the bricks, right, and the internal walls in the house. So same way, this is a complete cytoskeleton where there are fibers which will be keeping the cell intact. Cell is not hollow, and all these structures, they are not moving into thin air, right? There is a complete fibrous meshwork in which everything is connected. So these vesicles are also connected to those walls, right? Now action starts. Action potential has arrived, right? So things were like this, right? And we say action potential, right? Action potential. That means because of, as we were discussing, because of neurotransmitter, because of some, right, some voltage, right, some voltage, and it led to, first thing first, opening of voltage gated, right, because of action potential, right? So when action occurs, all the synapses will break at a time. Well, yes, everyone will start acting, but see, this is a natural phenomena, right? So calcium will be taking part, so you will get the answer that what would happen, right? Actually, yes, depending upon the action potential, depending upon the calcium which is entering, depending upon those mechanisms which will be even inhibiting it, stopping it, right, one by one, say they will start breaking. But yes, we'll talk about this point also. Just let this point complete, right, the point which we are about to discuss. So opening of voltage gated calcium channels, right, calcium channels, they open. Now it is not so that just calcium has entered and dhum dhum everything starts. No. There is a system. And what system is that if we say that this is the cell, right? And now we are make just drawing these vesicles, right? And we are just writing acetylcholine, acetylcholine, acetylcholine. So these are all vesicles connected, right? Connected with the cytoskeleton, right? All of them. So voltage gated calcium channels, right? They have opened because of the action potential, right? Because of that action potential. So calcium enters, right? So that's the calcium, and now calcium has entered. This calcium will play few major roles. So coming back to this, when we were 5, 6, step number 6, and then we were understanding that such synapsin is there, right? So we put it as number 7 just for the understanding that that is also one of the important point, right? And then the action potential arrives. That's our 8th point, and then it leads to opening of these calcium channels, right? And obviously these calcium channels, because it is action potential, so it would be voltage gated, right? Voltage gated action potential. Yes, it would be almost same for all the neurotransmitters. Very true, right? This process would be almost same for the neurotransmitter, but it is then the neurotransmitter will decide. Say if the GABA is released, so then GABA will say कि मेरा काम तो inhibition का है, so then it will be inhibited, right? So who it will act on what would be released, right? But for the acetylcholine, most it is exactly like this. But for other neurotransmitters, yes, there would be some similarities, but at the same time there will be few differences also. Coming back to this voltage gated, right? So it will open. So this is like calcium has entered, calcium entered. Now what this calcium will do, right? The first thing what calcium will do is it will, these are synapsin, synapsin, correct? So let's say, okay, let's break these synapsin. So this is 11. Synapsin breaks. And when synapsin breaks, it is called as the mobilization of vesicles. Chalo. So now vesicles, they are ready to travel to come to this surface, because it is this surface which has to be broken, and then it has to go into the synaptic cleft, right? It has to go into synaptic cleft, and in synaptic cleft they will release, because then the process would start. Ke chalo, now the action starts. But here comes one more character, and that is called as the snare protein, right? You got it? So this is 11, this is 12, and now our situation is that this is our cell and our free vesicles, right? Free vesicles, they are telling that now there is no synapsin, so we can travel towards the surface. So here comes snare, snare proteins, right? Now, do you remember we talked about these snare proteins before some time, right? Snare proteins. So for the sake of ease, what I've done is I've kept that file open. Yes, right? So the same, this file, you have got this file. This is that neuromuscular junction file, right? NMJ. So that's the snare protein, which is SNAP receptor protein. And SNAP receptor protein, they were like soluble NSF attached protein. NSF was ethylamide sensitive factor, but in nutshell, that's right. It was also called as the vesicle protein or VAMP, that is vesicle attached membrane protein. And very true, that protein is none other than the synaptobrevin, right? That is the synaptobrevin. Now to this we can add something even more also, right? We'll add it. But say this synaptobrevin, there are other proteins like syntaxine, right? Or the syntaxine, or the or the syntaxine, or there is one more protein which is called as SNAP25, right? SNAP25. These are all surface proteins, right? All surface proteins which are present on the surface. So let's write it over here. So this is the surface, right? This is the surface. On this surface, on the membrane, we are got syntaxine and SNAP25. SNAP25. They are snare protein, and on our vesicles we have got over here, here, right? And that is our, very rightly said, synaptobrevin, right? Synaptobrevin. Now these proteins, right, they are tremendously attracted to each other, right? So immediately they will start traveling. They will start traveling, right? So where are we? Where are we? This was number 11, that is synapsin breaks. So that leads to mobilization of our vesicles, right? Sort of, yeah, that protein is a sort of transporter, but this is for the membrane disruption, so we can't tell it transporter. As such, acetylcholine would be released. So this is 12, and then say point 13 is our synaptobrevin, and we can say that this is on cell surface or the cell membrane, right? So that one would be 14, right? And they start traveling. So this one would be number 15. They will be coming near to each other. Now there is one more character to be added into this, and this one is synaptotagmin. Synaptotagmin. We give it number 16. Right? As such, let's change the number, right? Let's give it the number 13, 14. Let's give it this 15 and 16 here, right? This is acetylcholine, 16. What synaptotagmin is? Synaptotagmin is a calcium sensor. Right? This calcium sensor, which is present in vesicles, it keeps that yes, sufficient calcium should be present, then and then I'll allow these vesicles to move towards the surface, right? So it is a calcium sensor, and sufficient calcium be present. So that's the synaptotagmin. Sufficient calcium is there, and they start traveling, right? And they start traveling, and finally at this particular junction over here, right, there is formation of what's called as the snare complex, right? There is formation of snare complex. That's the place where synaptobrevin, syntaxine, SNAP25, they all will come and they will form the snare complex, right? They will form the snare complex. That's our step number 17, right? Now at this for the snare complex formation, there is one more. It is called as the monk 18, right? That also helps. So this is like a monkey, right? So that monk, it is monk 18, helps. So he also jumps in and just helps in formation of snare complex. So surely we give it, say this is 16, right? Let's give it 16b, right? 16b. So it helps. As such, so much of literature is there, and yes. So now snare complex is ready. Snare complex is ready, immediately that cell wall is broken and our guest of honor comes out, and that is the release of acetylcholine, right? Released. Choline is released. So this is number 18. Right? So it is released. So now this is our synaptic cleft, right? Monk 18 helps in formation of snare complex, right? It helps in snare complex formation, right? It helps in this. So acetylcholine is released, right? That is released. So this is the synaptic cleft. ठीक? Now this acetylcholine, as it is released, it will start working on, right? It will working on nicotinic or muscarinic receptor. True. So if we really draw it like this, and let's say that's our ion channel, right? That's our ion channel. So which will be permitting the transfer of ions. So that would be our nicotinic receptor, right? It would be our nicotinic receptor. So if it is acting on that, it will open those channels. See, there is a type of it is not type of, it is exocytosis. And in fact, my mistake, I missed to tell you that, right? ACT, this is very good. It is called as exocytosis. You are million% right. That is the process of exocytosis. Thanks. Right? Very right. So acetylcholine is released by the process of exocytosis. Absolutely perfect. So this is released. Now, there would be nicotinic receptors which are in the form of channels. So it will open those ion channels and there will be transfer of ions, right? Or there could be that 1, 2, 3, 4, 5, 6, 6, 7, right? Or there would be GPCR, G protein coupled receptors, right? Which are the sort of muscarinic receptor, right? These are muscarinic receptors. So their function would be completely different as we discussed. So basically, whether it will be acting on the muscarinic receptor or it is acting on nicotinic receptor, but the final aim is it will lead to what's called as the cholinergic effect. Effect generated by acetylcholine is called as the cholinergic effect, right? Right? It is called as cholinergic effect. Right? That's why we discuss this thing in slightly advance, right? That what happens to nicotinic, what happens to muscular, right? This was for neuromuscular junction, right? That's right. Muscarinic was for our must heart, right? And that was leading to slowing down the heart, etc. So you know that. So that leads to this effect. So when it is 18, so collectively we write 19, right? So whether it is nicotinic or muscarinic, but we get the cholinergic effect. But we don't want that it should keep on working on and on and on, right? So there has to be a control. That control, it comes over here. This particular acetylcholine, it has to be broken down, right? We need to break it down. Otherwise, so it is going and it is acting on those receptors, right? But it has to be broken down by acetylcholine esterase, right? Acetylcholine esterase. So it will simply separate acetyl, that is acetate, plus the choline, right? It will separate both of them, right? And then this will be taken back. So here, that's the 19, acetylcholine esterase, that's our 20, and this choline is taken back and it is called as reuptake. That means it is recycled. It is taken back, right? So 21, that's the point number 21. Meantime, those vesicles, right? Those vesicles, when they are broken, right? There is snare complex, right? That was the snare complex which was broken, right? Snare complex. This snare complex, when it is broken, there are snare proteins which are moving. So now this particular complex, right? What happens that even the vesicle is to be recycled. 21, 21 was, acetylcholine should not work endlessly, right? So this acetylcholine should be broken, right? It is what is called as the degradation, right? This is called as the degradation of acetylcholine, degradation. Otherwise, it will keep on working on and on and on. So this is degradation. So it is broken down into acetate and choline, and then this choline is taken back. It is the reuptake. It is taken back into the cell so that it can be recycled. It can be used again, right? This is what is also called as the rate limiting step. So if acetylcholine, you do something like in which break, you just break this cycle, that is release of acetylcholine esterase, if that is broken, right? If it is broken, yeah, it is reabsorbed. But if acetylcholine esterase is blocked, that means acetylcholine will remain present. It will keep on acting on more and more number of receptors, right? And if you increase the concentration of acetylcholine esterase somehow, so it would lead to decrease in the effect of acetylcholine because it would be immediately broken down and that they'll be taken back. So there'll be less chance of they acting on it, right? Similarly, the snare complex, right? All those proteins, they are also that this snare complex is formed, right? It has to be broken, right? So for that, NSF, these are the proteins, NSF and SNAP proteins, they come into picture, right? Now these are what? These are also snare proteins, right? So just remember that these snare proteins, they are inside and outside, so break, they break this complex, right? And thus all the components, they are broken down and then vesicle, right? It is reabsorbed. So that nothing is left out. Everything is utilized. That even those broken ends, they are also utilized. Interestingly, that acetylcholine, right? It never stops. Yeah, it never stops. It keeps on release. It keeps on getting released, right? It is released constantly. Constantly it will be released. But the quantity is less, right? Quantity is less. This quantity increased when action potential comes into picture. When action potential comes. But otherwise, in small doses, it will keep on releasing, right? So that's how they really work. Now see, let's say this is blood brain barrier. Synapsin network, yeah, synapsin networks, it happens this way, that synapsin works in presence of calcium, right? Synapsin breaks, right? So there is mobilization of vesicles. The moment calcium is depleted, right? If calcium goes, that synapsin, it forms again and it again starts capturing those vesicles, right? Synapsin is broken only in presence of calcium. So as the calcium level increases, more and more number of synapsin will break and those vesicles, they will keep on moving. So it is like a batch, right? It is like a group. Say, let me draw it in here. Right? So if we draw it like this, right, and all of these are vesicles, right, all of these are vesicles, and right, they all are connected, right. So when the calcium enters, right, these vesicles, right, they are mobilized. So step by step, right, they are mobilized. It's not so that all of them they will be acting together, because over there also there is a mechanism of calcium which is taking part in the break of synapsin. So typically, right, this is broken, right, so they are released. So when the calcium level stops, so fine, rest of them they are not broken, right, they are released, they are again connected to cytoskeleton as more. Once again, when the next action potential comes, again the new batch would start breaking, right, because those who have reached over here, so bas that is their end of life, right, they have released their acetylcholine, right, and their function is over. Bas now these proteins, so they are to be absorbed, right, so their function is over, through, no more. Esterase comes, then yeah. So when this acetylcholine esterase comes, then calcium is depleted from vesicle. Well, calcium would be depleted only by one way. That is when the action potential stops. When the action potential stops, if AP stops, so then calcium channels will be blocked, right? And when the calcium channels are blocked, so that's it. So now this entire process would stop, right? The entire process would stop. So when we'll be discussing about say various drugs, so this is how they will be working on all these various steps, right? Whether, and that is how we define whether we want to continue with acetylcholine or whether we want to stop it, right? Everything will be on, it will be acting on one or more steps, right? Even the toxins, right, they will be acting on one or more steps. That's the reason that all these steps they are so crucial. Acha, chalo, because we have talked about it, let's discuss so that this concept becomes crystal clear, right? We were talking about the receptors, parasympathetic receptors, right, and we said the muscarinic, right, muscarinic. So muscarinic receptors, they are GPCR. You know what is GPCR, right? G protein coupled receptors or serpentine protein or 7-pass receptors, all same, right? I'll just write 7 so you know that. 7-pass receptor. Nicotinic receptors, right? Nicotinic receptors, because it is the nicotine substance. It is in the intracellular fluid. Which one? Calcium? Yeah, calcium would be in ICF. That's right. That's right. Because vesicles, so they are just forming that acetylcholine. They are the bags of acetylcholine, right? So this nicotinic, right? They are all ion channel receptors, right? So we can say these are like ligand gated. Obviously, it has to be ligand gated, right? Ligand in this case is our acetylcholine, right? Ligand gated sodium ion channels, right? Sodium ion channels. Now, to clarify it further, we have to divide this into two parts. One which is muscular. So we have got, we'll write this thing as NM, nicotinic muscular, right? And second one would be neuronal. So we'll call it NN, that is nicotinic neuronal. So this muscular, obviously they will be going to neuromuscular junction, NMJ, right? And that is what leads to muscular or the muscle contraction, right? This is quite good. Neuronal, that is bit special, Right? Let it take you to another page. That is a bit special. It will act on adrenals, adrenal glands. It will act on ganglion, right? And it will act on central nervous system. It will act on the central nervous system. So over here, this acetylcholine, when it acts on adrenals, what really happens? It will lead to increased synthesis of noradrenaline, right? So it will increase synthesis of noradrenaline, right? Or you can say norepinephrine, right? One and the same. This will be making epinephrine, right? Epinephrine. Now, epinephrine is what? Epinephrine would be released into plasma, right? So you can say these are like a mechanisms. So this is like a hormone, right? So it's like a hormone. Anything which is related directly into the plasma, into the blood, chemical, that is called as the hormone. Right? It is not requiring any of the neurotransmitter. It is releasing directly into the blood, right? That is the adrenal, right? So from adrenal cortex, this thing is removed. That is the cortisol, right? So norepinephrine converted to epinephrine. Similarly, when it will be triggering, it will be talking to ganglion. So it will be regeneration of action potential, so that things are carried further, right? And in CNS, when it is acting, so it stimulates and can lead to seizure, right? Convulsions, seizure. So that's how this, say, neuronal receptors, they will be working. Now when we talk about, say, muscular, right? So these muscular are also divided, right, into M1, M2, M3, M4, M5. That's right. That's right. It controls, right? It controls all these mechanisms. This I'm just think of that where acetylcholine is working, right? Where exactly it is working. So say this muscarinic, right? When we are telling this muscarinic, right? So if you go for the subtype, then you have to write it like odd and even. You have to write it like M1, M3, M5. These are the subtypes of muscarinic, and M2 and M4, right? M1, M2, M3, M4, M5. Seizure means convulsions, right? So this one and this one, right? Yeah, norepinephrine release epinephrine in presence of cortisol, right? That is from the adrenal cortex, right? From the adrenal cortex, cortisol is released, cortisol, and that cortisol will lead to norepinephrine to epinephrine. Achha. In muscarinic, again there are 5 types, right? So these M1, M3, M5, they are excitatory, right? And M2 and M4, they are inhibitory, right? So this one is like that GQ, and this one is GI, right? In receptors, we talked about it. And where do we really find them, right? Where do we find them? Very classical, right? M1, M1. GS is stimulatory. GQ is exactly QISS, exactly, right? So M1, right? It will be acting on CNS, right? And what it will do, it will improve cognition, right? See how effectively this acetylcholine actually works on so many things. So when what cognition is focus, right? Something which is lost in Alzheimer's, right? Something which is lost, that focus goes, memory goes, right? Person forgets. So what if this M1 receptor, which is acting on CNS, right? Which is acting on CNS, and this particular fact, if we take M1 stimulant, if we can figure out a drug which is stimulatory to M1 receptor, so then that thing can be helpful in Alzheimer's, correct? It can be helpful in Alzheimer's disease. And one of such drug, right? Preferably, remember its name. It is called as the taclifencine. It is taclifencine. Very positive results. But this is M1 stimulatory. Because when M1 is stimulated by acetylcholine, and M1, which is a muscarinic receptor, and M1 is stimulatory, right? When it is stimulatory, that means it improves cognition because it is affecting. Memory lost is lost. How it comes again? Well, we'll discuss about the memory, because memory, there are many. Say some memories, they are short-term memory. Short-term memory is now the cell phone has come, but otherwise, if you remember, you have written someone's number in diary, you say that okay, this is the number, and you dial that number, and after that you forget. Completely short-term memory. Your name, which is into permanent memory or super long-term memory, which you never forget, right? Any of the insult which touches the heart, right? which touches the heart, right? It is long-term memory. Acha. So this is M1, right? M1 will also act on, it will also act on GIT, right? So when it acts on GIT, there would be increased secretion, right? It would be increased secretions, right? So this is how M1 is working. On to the M3. Again, M3 is what? Stimulatory, right? This M3, this is most common, right? Remember, all these are in fact questions. Huh? These are questions. This one is a question. This one is a question, right? All. Yeah, this is so definitely a question, right? Then yeah, this one is a question. This one types GPCR, right? These are sodium ion channels. All these are questions, right? So when we'll be conducting exam, so we can ask these things as questions, right? And many times I disclose that these are the questions, and then I ask them in exam. The intention is you remember it. Acha. So M3, which is this is most common. So this is also in GIT. Stimulation of GIT, this is positive, so it will lead to increased contraction, right? It has to. It will lead to increased contraction, no doubt. When it will be acting on glands, so what will it will do? It will lead to increased secretions, right? Simple as that. It will lead to increased secretions, right? Muscles. So when is the next exam? I don't know. Right now, we do have quite time hai abhi, right? It's not in near future. Still, I have to check. Huh? But definitely, this is December. It comes somewhere in the month of March or April, or even a bit late. Because of COVID, it was pretty delayed, right? Now in bronchi, right? Bronchoconstriction. Acha, our exam. Okay, I straight away think about, because I answer so many emails about the next exam. So I always feel like next exam means you are asking for the national exit exam. Acha, our exam. Just ask this way, our exam. We'll plan it. We'll plan it. Then this weekend. So bronchoconstriction, right? Because muscles, they are constricting, so they constrict. YouTube, I got it, right? Acha. Then detrusor muscles, right? Detrusor muscles, urinary bladder, they will contract. Yes, because parasympathetic, so that would lead to increased contraction. So in rest and digest, person would feel like, chalo, let's go to washroom, right? Similarly, those circular muscles of iris, right? Circular muscles of iris. So that will lead to constriction of pupil, right? That will lead to constriction of pupil. So meiosis, because this is rest and digest, aram, right? So this is constriction of pupil. So pupil is not dilated, but pupil is small, right? This is small. Who is responsible for this? For this, olfactory uptake oculomotor. This is because of, yes, cranial nerve number 3, right? That is oculomotor. See, this is where those numbers which I told you, right, they come into picture, right? So that is cranial number 3 is coming into picture. Then this, as we said, M3 is most common. So M3 would be acting on blood vessels, right? It will be acting on blood vessels. And in case of blood vessels, think it like when the smooth muscles they contract, it will lead to vasoconstriction, right? Yeah, 379. So this, it will lead to, so smooth muscle constriction, right? Smooth muscles constriction, right? So that will lead to vasoconstriction. Correct. Similarly, when it will be acting on, say, our endothelium, right? Endothelium. So in endothelium, it will lead to increase calcium release, right? So this thing will stimulate what is called as the calcium dependent enos, right? It is called as the calcium. This is very, very important. So why nitric oxide is taken? Because when many of the gym people, right, they take nitric oxide because it is acting on this vascular endothelium, the lining of the vessels, right? So when the lining of the vessels, they are affected. So this calcium dependent, right? Calcium dependent enos, that is endothelium or the endothelial nitric oxide, right? Nitric oxide synthetase, right? Or enos. This is affected. So this it leads to increased nitric oxide, and that increased nitric oxide leads to vasodilation, right? It leads to vasodilation. So gym people, in many of the pre-workouts, if you'll see, they'll say that this releases nitric oxide, so that when the nitric oxide is released, it leads to vasodilation, and that leads to prominent vessels, right? Prominent vessels on the body, right? Especially the arms and the legs, especially the arms, right? So that leads to vasodilation, right? And this is pretty powerful. This is pretty powerful. Acha. Then comes our M5, right, which acts on CNS, right? And it can lead to that seizure, etc., what we talked about, right? Then M2 and M4, right? They are inhibitory. So this is acting on CNS, but it is inhibitory. This is also inhibitory. So when M2 acts, right? So for the presynaptic, see, these are all negative feedback mechanisms, right? So in presynaptic, it can decrease acetylcholine release, right? This can decrease acetylcholine release. So it is like a control mechanism. But most important is in red, that heart, right? Want to draw it like this? No problem, right? Because it acts on SA node, right? SA node. Sinoatrial node, which is our pacemaker. So it inhibits seizure, means that delivery seizure. That is uh, those when the convulsions, when the body shakes, right? Convulsions. That is what is called as seizure. Means grand mal seizure, petit mal seizure. There are types, but seizure you can say it like convulsion, right? Convulsion. Achha. So SA node, shakiness, yes. So current is inhibited, right? And at the same time, it leads to diastolic depolarization, right? Just remember, it leads to diastolic depolarization. So now the heart relaxes, right? This leads to decreased heart rate, because when the diastole is extended. Diastole means when the heart is relaxing. Systole is when heart is contracting. When the diastole is you are giving more time for diastole, so that means heart is relaxing, relaxing, relaxing. Let chalo blood aane do aaram se, and then contract. So it decreases the heart rate, right? It decreases the heart rate. Similarly, this M2, right? M2, it acts on AV node, because AV node is like our backup, right? In case if anything happens to AV node. So here also it inhibits, right? L-type, what it really inhibits is L-type calcium channels. That's what it is said. L-type calcium channels, they are blocked, right? And obviously the myocardium, the muscles of heart, so they are also affected. So which leads to decreased contraction, right? It leads to decreased contraction. Now this particular effect, right, this is not very high, right, because there are other mechanisms. So this is a minimal effect, but even if you want to figure it out, it is more prominent on atrium as compared to ventricle, right? That is the myocardial effect. It is more on atrium as compared to ventricle. Now on Purkinje fibers, there is not much of the effect. So that's how all these are distributed, right? So this is about our full synaptic transmission with the involvement of all receptors and all the intricate steps which are in between, right? Let's go for a super high speed revision, right? Oh, almost 2 hours have passed. 1.5 hours. 1.5 hours. Joint effort, right? It is like a joint effort of so many people, right, who really discovered all these things. But still it is, the thing is like salute to God, right? How these things, they really work millions and billions and trillions times in the whole lifespan, and then how the entire thing, it keeps on changing as per the circumstances, right? Such an amazing thing. Chalo. So let's start with our high speed division: body, CNS, and the autonomic nervous system, parasympathetic system, sympathetic, enteric. This is rest and digest, and neurotransmitter would be acetylcholine, right? Fight, fright, and that is the enteric, which is into the GIT. We have the cranial nuclei 37911 for the parasympathetic system, and S2, S3, S4. Point to remember is that S1 is part of the thoracolumbar. There is a sympathetic system. My pleasure. Then limbic system, all the inputs from eyes, ears, right? All various inputs to the hypothalamus, and the hypothalamus, in which we understand it by this figure, right? This is just a schematic figure so that we know that anterolateral part is parasympathetic and the posteromedial part is sympathetic for the memory, emotions, learning, cognition, right? Cognition. When it is said that females, they have got better cognition power, right? Their cognitive ability is much fast. They are able to focus on multiple things together, right? So here it is our the ganglion, right? This ganglion, the fibers before that they are preganglionic, and fibers after that they are postganglionic, and the neurotransmitter is needed. Exon, somatic, exon dendritic, but there are a number of neurotransmitters, so many neurotransmitters, and still the work is going on and on, right? Class amino acids, this is like glutamate, GABA, which is inhibitory, right? Alpha amino butyric acid, and the glycine, right? The fundamental difference, glutamate, which is stimulatory, right? It opens sodium channels. Sodium comes in, so obviously when sodium comes in, so it leads to depolarization. Things are going plus, 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 diagiri, right? So happy, right? When it comes to GABA, GABA opens chlorine, right? So it's chloride. When the chloride enters, right? Chloride enters, so things are negative, negative, negative. So it leads to repolarization. So no chance of AP, right? Or the action potential. Nucleopeptides, endorphins, happy, extremely monoamines, epinephrine, norepinephrine, dopamine, serotonin, and histamine. Serotonin, it is like for a pleasure, which is only for short term, right? This is in itself is a very superb topic. That what happens to serotonin and what is dopamine? What are they really doing? Right? What happens when the endorphins are released? Endorphins, usually they are the hormones or the these neurotransmitters which are for the pleasure, right? Serotonin, it is for very short term pleasure. Anyway, coming back to acetylcholine, right? Ester of cholines, and this is where in high speed we saw that those vesicles, they are into those carrying neurotransmitters, right? And they come out by exocytosis, and it is the action potential which triggers calcium channels, and calcium enters and the whole process starts, right? So this is acetylcholine, nicotinic and muscarinic receptors. Nicotinic, which will be acting on NMJ, and they contract muscarinic cardiac muscles, slow down the heart, right? And especially they work on SA node, is also present in depression. That's right. That's right. So acetylcholine, the window is very small for few milliseconds, and it is degradation of acetylcholine and reuptake by astrocyte, so that it takes it back by the process of diffusion, and it is taken back. This was the conceptual thing, that the acetylcholine in all this ganglionic fibers, whether it is parasympathetic or sympathetic, it is just acetylcholine. Only the target changes when it is parasympathetic or sympathetic. So in target acetylcholine receptors, they are muscarinic or nicotinic, and sympathetic targets are alpha and beta receptors, right? Obviously there are subtypes, right? Renal blood vessels, dopamine, vasodilation, diuresis. Over here cell head soma, here the peptide neurotransmitters, they are there, high molecular weight, they are synthesized, they travel to exon, reach to these terminals, and over here in the form of vesicles, right? They are packed and then they will be released. In acetylcholine, choline is coming from the food, right? This is the conceptual part. 6 carbon glycolysis, 3 carbon, 3 carbon pyruvate, 1 carbon knocked off by pyruvate dehydrogenase, 2 carbon, that's called as acetyl, right? But one end is sticky, so this coenzyme, that sticker comes and sticks on it. But this coenzyme A, it has got one reactive area, and that is where it calls for choline. So that's how acetyl CoA and the choline, they lead to formation of acetylcholine, right? Journey begins mitochondria. This happens in mitochondria. Acetylcholine and comes formed. Choline comes from outside, and that is by sodium choline co-transporter, right? Choline comes in, acetylcholine formed. Immediately it is formed into the form, these vesicles, they enter, right? Hydrogen comes out, all the vesicles, they are now loaded, packed by this synapsine, right? Synapsine is binding, right? So that all of them, they are into the form of clump, right? So that they are not allowed to go onto the surface and release acetylcholine. And then comes the action potential. It leads to opening of voltage gated calcium channels. Sufficient calcium should enter. That's why we drew more number of calcium channels. Now when they enter, calcium starts working inside the calcium, right? First it breaks the synapsine. That's what called as the mobilization of vesicles. So all of them, they now start floating, right? But then comes our snare protein. Snare protein is like they are like gangsters, right? There is a big gang, right? So this is the synaptotagmin. He is like a driver, right? He is controlling for the bank robbery, right? He is controlling that how much calcium be present, then you start the action. Sufficient calcium, so then he says, chalo, start the action. So here is synaptobravin, and on the border, right, on the edge, these are the syntaxine and SNAP25, they are sitting, right? They come near each other, breaks, and then acetylcholine is released. Meantime, this monk 18, right, a small monkey, he also helps, right, in this entire process. Snare complex is formed and acetylcholine is released. The process, what is called as the exocytosis, right? Now this acetylcholine should not keep on working, so it is degraded, right? Acetylcholine esterase by acetylcholine esterase, acetyl plus choline, and it is taken back. But this acetylcholine, it will be acting on muscarinic receptor and it will be acting on nicotinic receptor, whosoever is present, right? Wherever it is releasing. So in case of nicotinic, these are the ion channels, right? Obviously the ligand gated, because this is like a ligand, and the muscarinic, which is GPCR, it acts, and finally they will be leading to cholinergic effect. Again, where exactly are they? In the eye, so then it will lead to pinpoint pupil. If it is into the heart, so that will lead to slowdown of the heart. If it is into GIT, that will lead to, right, good contraction and the digestion. So these vesicles, once again, these snare complex is formed, and which is allowing ke chalo jao jao acetylcholine jao jao. So this snare complex is broken, right, by NSF, right, and the SNAP. These are also snare proteins, right? They say, no, no, don't do this. Snare complex is broken. Vesicles, they are all reabsorbed. They go back. Acetylcholine is released constantly, right, in small quantity, right, in small quantity, right, always, but increased when action potential comes. This is where we talked about that synaptin part. Muscarinic, nicotinic, right? These are GPCR, 7-pass receptor. Nicotinic, it is ligand gated, obviously ligand gated sodium ion channels. Muscular and neuronal, NM and NN. Muscular will be acting on NMJ, and neuronal will be acting on adrenals, ganglion, and CNS, which will lead to increased synthesis of noradrenaline and finally the epinephrine, right? This is regeneration of action potential onto the ganglion, and this can lead to seizure or the convulsions, right? Convulsions or the shakiness. What you said is very right, shakiness, right? Muscarinic M1, M2, M1, M2, M3, M4, M5, but M1, M3, M5, excitatory, M2, M4, they are inhibitory. Right? M1 acts on CNS, improve cognition, and that is teclifencin, a good drug. Right? GIT acts on glands, leads to increased secretion. But M3, which is the most common, and you must remember, this is GIT, increased contraction, glands, increased secretion, bronchi, more breathe is not needed when we are resting, so bronchoconstriction. Detrusor muscles, yes, now is the time, there is no need to fight with anyone, so washroom can be visited, right? Iris, no need, we are not running anywhere, right? We are just relaxing, so pupils will be constricted. So that is called as the meiosis, right? Cranial nerve, 3 oculomotor involved. Blood vessels, smooth muscles, they constantly lead to vasoconstriction, right? No need to all those dilated vessels and fight, right? But when it comes to endothelium, there is increased calcium, right? And calcium dependent enos, that is endothelial nitric oxide synthesis. So this leads to increased nitric oxide and can lead to vasodilation when it acts on endothelium, right? This is for the smooth muscle constriction. M5 CNS, M2 presynaptic decrease of acetylcholine, right? And finally on SA node, right, decrease current, decrease diastolic inhibition of diastolic depolarization, so heart rate drops. And AV node, L-type calcium channels are broken. Myocardium again leads to decreased contraction, but it is minimal. Atrium is more as compared to ventricle, right? So this was our today's topic, right? Now, what we'll do tomorrow is we'll pick up something, we'll finish muscle, right? We'll finish the muscle part. Just one session left out for muscle so that we see the entire muscular process, how it contracts, and then we'll move on to the completely new area. Tomorrow, same time, I don't know. Yeah, that's important. Is morning, because see, again, very few friends are there, right? So tomorrow we can take even in the morning part at 11 o'clock. Is it fine? Or for tomorrow, send me a mail, right? So we'll decide at that point. Morning was better. So we can take morning. Morning 11 o'clock. Morning is fine. Chalo, done. Very nice. All right, done. So thank you so much. Right, and see you tomorrow morning, right? Just after a few hours, right? Take care and see you. Bye bye.